So for today's talk, Sarah is going to talk about her use of stem cell technology and how she uses stem cells to model Parkinson's disease in a dish. So without further ado, Sarah, please take it away.
Thank you. Thanks to all the other speakers for their great talks so far. I'm delighted to follow up them. Can I just check that everyone can see and hear me okay? Maybe a thumbs up. Excellent. Also, cheers. I feel like I'm the only one drinking, so cheers to whoever else is having a pint tonight, and hopefully next year we can be back in the pub. So just to quickly introduce myself, I'm a postdoctoral research fellow in Trinity College Institute of Neuroscience. And I don't want to go too in depth into my background in science, but just to any budding scientists following the talk today, listening to this recording in future, or if you have any budding scientists at home, I very much just followed what I was interested in. I loved science in school, performed quite well in science for junior cert, so then studied biology and chemistry at Leaving Cert level, and again they were my strong subjects, so it was kind of a natural progression to apply for science at university level. And I hadn't really considered what I would do after my undergrad until my final year, when I carried out a research project and got to do some hands-on lab work and loved it. So I knew then that I definitely wanted to stay in the lab. But just to emphasize that I don't feel like I'm fulfilling a childhood dream, I just kind of followed my passion along the way and almost fell into it a little bit. But the current research project that we're working on, and maybe I'll give a shout out to the Health Research Board for funding it, focuses on Parkinson's disease. And in order to do that, I'm modeling Parkinson's disease in a dish. So I'm going to mainly talk about that today, but I wanted to start by maybe just giving a little bit of a background on Parkinson's disease itself for those who are not familiar with it. So Parkinson's disease is a neurodegenerative disorder. It is the second most common neurodegenerative disorder after Alzheimer's disease, and the leading neurodegenerative disorder associated with motor dysfunction. So the main outwardly present symptoms of Parkinson's disease, which also happen to be the clinical criteria, include that resting tremor, so shaking without any intention, slowness of movement, unstable posture, altered gait, so maybe changes to how a person walks, and rigidity and maybe difficulty initiating movement. There are also some non-motor symptoms which are important to note as well. So people living with Parkinson's disease can also suffer from anxiety, depression, and disturbances to their sleep. So really it's quite nasty, and you wouldn't wish it on anyone. To look at the level of the brain, what's actually happening is a cell population in the midbrain. So this is maybe the middle of the brain. If you sliced right down through the nose and looked in sideways at the brain, you can see in the pinky purple right in the center there, the substantia nigra of the midbrain. And then looking as if you had sliced the brain from the forehead through to the back of the head, you can see the same substantia nigra actually has this wing shape. And within that substantia nigra is a population of cells called dopamine neurons. And in Parkinson's disease, these cells die. And their function in the brain is to produce dopamine, which is then sent to all of these other regions of the brain that are important in movement. So then it's no surprise that if there is a loss of dopamine producing cells and then dopamine itself, that you will get these motor deficits associated with Parkinson's disease. There are a number of treatments currently available for people with Parkinson's disease, but they only target the symptoms. They only aid management of symptoms, and they're not really effective for everyone. So there is maybe a small cohort of patients where the current therapies are effective, but a scary number where it's not. And none of the therapies available to date actually replace the cells that are lost or even stop further loss of cells. So the disease in itself will continue to develop, and further degeneration will occur. So really it's necessary for a better understanding of what's happening in the brain in Parkinson's disease, and then to hopefully find better therapies down the line. So we want to have effective models of Parkinson's disease in the lab in order to better understand what is happening with the cells in the brain. And a lot of research to date has made use of animal models. I don't know if that will come as a surprise to some of you, and maybe it won't. But there are a lot of differences, I'm sure you can imagine, not just visually, but genetically and behaviorally between humans and animals. So we wanted to have a human model to maybe better model Parkinson's disease in the lab. So I'm going to just give a little bit of an explanation as to what stem cells are, because we are in fact aiming to use stem cells in our model system. So we all started from a stem cell. The fertilized egg is what we would call a totipotent stem cell, because it can develop into all the cells in the body, as well as the placenta and the umbilical cord and the embryonic sac as well. And then this will further differentiate into the little embryo. And within the embryo, you have this bundle of cells that are called pluripotent stem cells, because they will derive any cell type in the body. So neural cells, muscle cells or blood cells, and then the different types of immune cells, liver cells, all the cells. So we would like to harness that pluripotent ability of stem cells so that in the lab we can differentiate whatever cells we want. And there is this fantastic technology that was discovered in 2006 that adult cells can be reprogrammed to become pluripotent. So essentially reversing this process. And that was such a breakthrough in science and research modeling that the lead investigators were awarded the Nobel Prize in Physiology and Medicine in 2012 for their work on this. And just to put that in perspective, I guess it's the Nobel Prize is like the Oscars in the science community. It really is the pinnacle of awards and a fantastic achievement. So how do we go about reversing this process and generating what we would call an induced pluripotent stem cell? So we need to start with a differentiated cell. And what we actually do is start with a skin cell, because it's easily accessible. It can also be carried out by starting with maybe a blood cell, but some people might consider that a little bit invasive. It's not always easy to get a sample of blood, but everyone has got a little bit of a skin surface available to take a sample of skin. So I hope this picture doesn't bother anyone, but I just wanted to show what the skin biopsy looks like. So it's a 4mm dermal punch. This procedure is carried out by a medical doctor, so not by myself personally. This is the state in which I would get the sample from the clinic in the hospital. There's a little gadget called a dermal punch that actually can remove a little circular piece of skin. And we usually would take this from either the forearm here or the back of the shoulder, because those are typically areas of skin that are easily accessible, but also maybe don't get exposed to the sun too often. So that's perfectly ideal for us. So then back in the lab, when I've collected the skin biopsy, as actually happened today, I got a call about 3 p.m. We've got a biopsy for you. So I had to send my colleague running up to the Mater Hospital to collect it for me while I prepped everything in the lab. So this is just an image of what we do with the skin back in the lab. I would bring it into the culture hood. This is a sterile environment so that I can prep the biopsy and ensure that it's not contaminated with any kind of bacteria or fungi, which would grow in the cell culture, take all the nutrients, and the skin wouldn't thrive. So in order to avoid that, we use this sterile culture hood. So I've transferred the skin biopsy into this little Petri dish, and using a scalpel, I can cut it into 4 pieces and pop it into a tissue culture plate, like I've done here. After about a week, the skin cells or fibroblasts will start to grow out of the biopsy on the surface of the dish. So this is an image I took under the microscope. At the top of the microscope field, you can see the skin biopsy. It's really, really dark because there's layers upon layers of cells. But just at the very bottom of the biopsy, you can see that the fibroblasts are beginning to grow out. After another week or two, those fibroblasts will have coated the entire surface of that well. And at that stage, I'm able to move them to a bigger well to allow them to grow, and again to a bigger well, and again to a bigger well. So it's really quite easy to expand these cells. And another advantage is that they can be frozen and stored in liquid nitrogen for future use. Once I've got a population of skin cells, and I'm happy that I have some backups frozen, we can begin the reprogramming step. This is where we're at in our project at the moment. But briefly, what we do is insert embryonic genes. So the genes that are expressed in embryonic stem cells in the developing embryo can be inserted into the genome of these skin cells, and that effectively reprograms the cells or forces them back into a pluripotent state. And once they're in this pluripotent state, they have the ability to differentiate into any cell type in the body. So like the classic TV chef show, this is a batch of iPSC previously made, not from a Parkinson's disease patient. This is a batch of cells that we would use as control, so from an individual that does not have Parkinson's disease. But just to give you an idea of what they look like, they actually grow in little colonies. So this colony would have hundreds of iPSC, induced pluripotent stem cells, and then this defined border on the outside. And I took this image after a couple of weeks where the cells weren't looking great, and finally they looked happy. So I'm glad to show you it today. So once we've got a happy population of induced pluripotent stem cells, we can start differentiating them. So if you were to just leave them be, they will start spontaneously differentiating, and you might get random cell types from across the body. But because we're interested in Parkinson's disease, and because it affects the brain, specifically the midbrain, we want to make cells that are from this region, and we can do that. So thanks to developmental biologists, we know what signals are present in the developing embryo at the different times, and we can use this to our advantage in the lab. So in this figure, there's a representative image of the neural tube, which is the primitive central nervous system. So this section here would become the brain itself, with the forebrain being towards the forehead, the midbrain in the center where the substantia nigra is and what is affected in Parkinson's disease, and then the hindbrain, which be towards the back of the head. And then at the end of the neural tube, you would have the spinal cord, which is also part of the central nervous system. So it's possible to apply different concentrations of different factors at very specific timings to differentiate cells into any of these regions of the brain. So in order to make